Do you remember Dolly the sheep? This July marks 30 years since she was born. But what's been happening with cloning since then? For many people, Dolly was the first time science felt like it had stepped straight out of a science fiction film. A cloned animal, created from the cell of an adult sheep. When the news broke in 1997, it sparked headlines around the world. Some people were amazed, some were excited, and quite a few were absolutely terrified. Suddenly, everyone was asking the same question. If scientists can clone a sheep, what's next? But here's the interesting thing. 30 years later, the most important thing about Dolly isn't that she was cloned, it's what she taught us. Scientists at the Rosalind Institute took a cell from an adult sheep, transferred its DNA into an egg cell that had its own DNA removed. Against all expectations, that adult DNA didn't stay adult. It reset itself. The cell essentially started over and began developing into a completely new animal. That animal became Dolly. At the time, this was revolutionary. Many scientists believe adult cells had already made their choices, that once a cell became skin, muscle, or liver, there was no going back. Dolly showed that that wasn't even true. The instructions needed to build an entire animal were still hidden inside those cells all along. And that discovery changed biology forever. Over the following decade, scientists cloned cows, pigs, horses, cats, dogs, wolves, even endangered species. Today, pet cloning is a real business, and conservation groups are exploring cloning as a way to help preserve genetic diversity in threatened animals. But cloning itself didn't become the future many people imagined. In fact, some of the techniques used to create Dolly are far less important now than they once were. That's because modern scientists have access to incredibly precise genome editing tools like CRISPR gene editing. Rather than creating cloned animals to study genetics, researchers can now make targeted changes directly to DNA with remarkable accuracy. In many cases, it's faster, cheaper, and more precise. Yet Dolly's influence is still everywhere. The idea that cells could be reprogrammed helped inspire major advances in stem cell research, regenerative medicine, and even some of today's anti-aging research. And she changed something else too, public awareness. Before Dolly, genetic engineering was largely a niche scientific topic. After Dolly, genetics became dinner table conversation. People began debating cloning, biotechnology, designer babies, gene editing, and the ethical limits of science. Not everyone agreed. Some people were fascinated, while others were deeply concerned. But Dolly forced society to start thinking seriously about technologies that are now becoming part of today's everyday medicines. So 30 years on, Dolly's legacy isn't really about cloning sheep. It's about changing the way we think about biology. A sheep born in Scotland that helped launch one of the most transformative eras in modern science. Not bad for a farm animal.